In this lecture, we will be discussing how the three germ layers that were introduced in the previous lecture begin to organize themselves during the initial process of organogenesis. Gastrulation marks the beginning of a 5-week embryonic period during which body form is established and all of the major organs and organ systems are formed from the ectodermal, mesodermal, and endodermal germ layers. It is during this time period when these organs are most susceptible to teratogens, otherwise described as an agent or influence that causes physical defects in the developing embryo. Prior to this period, any disruptions in the normal developmental process typically lead to spontaneous abortion, often unbeknownst to the mother. Fundamental to the establishment of the general body plan is the organization of the mesoderm into a number of discrete cell populations. One population of cells is going to emerge from the primitive node, stay in the midline, and migrate caudally. These axial cells form the notochord, which is a solid rod that will grow to extend the full length of the embryo, as well as the precordial plate. Both of these structures serve important roles in the development of the nervous system, face, and axial skeleton. A second population of mesodermal cells is going to emerge from the primitive streak and migrate laterally to form both the paraaxial mesoderm as well as the lateral plate mesoderm. We'll be looking at these cell populations in a minute. For now, we will look at an additional population of cells which migrate cranial to the oropharyngeal membrane. This is referred to as precardiac mesoderm and will eventually give rise to the heart primordium. Now we get back to the non-axial mesoderm, meaning those cells that did not stay in the midline, but rather migrated laterally. 3 regions of non-axial mesoderm are established and can be recognized in these cross-sectional illustrations. The cells that lie just lateral to the notochord form the paraaxial mesoderm. With advancing development, the columns of cells are going to pinch off to form discrete tissue blocks in a fashion that resembles a string of beads. Each of these beads is referred to as a somite, and these somites are visualized in the bottom image. Somites are bilateral structures that are located along both sides of the notochord. The somites can be considered the building blocks of the body from the neck down, and we will consider their development in a future video. For now, it is sufficient to know that paraaxial mesoderm cells will give rise to the dermal layer of the skin. The epidermis, however, is derived from the ectoderm. Somites also give rise to all of the skeletal muscle cells of the body and most, but not all, of the axial skeleton, meaning the skull and vertebral column. These tissues, along with the epidermis, form the body wall. Together they are referred to as the somatic tissues of the body, and the word soma means body. Moving on to another region of non-axial mesoderm is the intermediate mesoderm. These are bilateral longitudinal columns of cells that are positioned lateral to but are continuous with the paraaxial mesoderm on each side. Intermediate mesoderm gives rise to most of the tissues of the genitourinary organs. A third population of cells develops just lateral to and continuous with the intermediate mesoderm and is referred to as lateral plate mesoderm. Following subsequent development, lateral plate mesoderm subdivides into a somatic layer, which remains in contact with the overlying ectoderm, and a splanchnic layer, which remains in contact with the underlying endoderm. The somatic layer contributes to the bones and cartilage of the appendicular skeleton. The splanchnic mesoderm becomes the smooth muscle, connective tissue, and serous membranes of the organs or viscera. The space between the two layers is referred to as the intraembryonic coelom. And the word coelom simply means cavity. It is termed intraembryonic because the cavity develops within the embryo and eventually gives rise to the thoracic and abdominal cavities. Now we consider how the ectodermal cells contribute to establishment of the overall body plan. One of the more significant events that occurs in association with the ectoderm is the process of neurulation, or the differentiation of neural tissue. The notochord, or rod-like structure that runs through the midline of the cranial-caudal axis of the embryo, gives off inductive signals that initiate the process of neurulation. In response to these signals, the ectodermal cells immediately adjacent to the notochord become columnar and form a distinctive neural plate on the dorsal surface of the embryo. The image to the right, depicting a superior view of the embryo, demonstrates that the neural plate is narrow caudally and considerably wider in the cranial region. The narrowed portion will give rise to the spinal cord, whereas the wider portion will give rise to the brain. Soon after neural plate formation, the lateral edges of the plate elevate to form neural folds. These folds then approach one another in the midline and eventually fuse to form the neural tube. The neural tube subsequently disassociates entirely from the overlying ectoderm. Neural crest cells are also formed during neurulation. We can see this population of cells located at the crests of the neural fold. As the neural folds begin to fuse, neural crest cells lose their adhesion to the fold, disassociate, and migrate to other regions of the developing embryo. Neural crest cells will give rise to sensory and autonomic neurons of the peripheral nervous system, neuroglial cells, or support cells of the peripheral nervous system, as well as melanocytes of the skin, the adrenal medulla, and connective tissues of the face and skull, as well as the dentin of the teeth. Here's another image showing many of the structures that we have been talking about. The midline mesoderm occurring here is forming the notochord, which, remember, is the signaling agent that induces the transformation of a population of ectodermal cells into the neural tube. Para-axial mesoderm, or that which is off the midline, is located here, forming the somite. Just lateral to that is the intermediate mesoderm, and then just lateral to that we have the two layers of lateral plate mesoderm, this being the somatic layer and this being the visceral layer. And then here we have the neural tube. Lastly, we consider the contribution of the endoderm to the overall body plan. The endodermal germ layer is going to contribute to the epithelial lining of the GI tract and its derivatives. In the flat, three-layered embryo, the endoderm forms both the ventral surface of the embryo as well as the roof of the yolk sac. Embryonic folding which is going to be discussed in the next lecture, converts the embryonic disc into a cylinder and the flat sheet of endoderm into a gut tube. This concludes our look at the early organization of the 3 germ layers in establishing a general body plan.